Welcome everyone for our next webinars in the Dana-Farber targeted protein degradation webinar series. And today we would like to try something different. For the first time, we decided to prepare Degron tagging workshops, the types of workshops where we invited three leaders in the field, and we would like to discuss techniques that I think it's a very important for our community. In terms of the degradations of this workshops, the tagging of the protein of the interest of the Degron tag is in the principle works that we are we will be discussing different types of the tags, where upon adding of the drug, induce the proximity between the tag and the E3 ligase, which leads to the ubiquitination and subsequent degradations of the whole fusion proteins. And we think it's extremely useful approach. First of all, to understand the protein functions. This allows us to deplete the targets within the minutes to hours, and gives an access to this all new field of the rapid biology. Our phenotypes can be achieved much cleaner and faster than the other techniques like RNAi and the CRISPR. And we can also assess the immediate functions for the essential proteins. And subsequently, we also think that the big advantage of this approach is that it's the easiest way to validate target as a degradation target for drug modality. And they think that the way that we discuss about those things, this gives the rationality that it's worth to develop the chemical binder to the protein of the interest, which subsequently can lead to the development of bivalent degrader, the PROTAC, or the molecular glue degrader.
But secondly, we wanted to effectively take a step back and assess all the different platforms that are out there, which is part of the reason for this workshop. And then another question came up is, what would be an ideal Degron inducing tag? So what would we like to have? And we basically were thinking that a tag needs to lead to a rapid and complete degradation, and emphasis on complete and emphasis on rapid. It definitely has to be a non-functional tag in the cells that you're effectively editing or tagging, and it has to preserve the native function of the protein. So that would put constraint on, for example, the size of the tag, or perhaps the nature of the ligand that is being used to control the protein fate. It has to also be an easy tag to tag endogenous proteins, either through knock-ins or otherwise. And then I think we could also take even a step back behind it and ask a question: does it have to be a protein tag? Can it be something else that would be able to induce like a ligand-induced control of protein fate? And finally, the second step, it has to be controlled. So the way current degradation tags are controlled is with addition of the ligand. So the ligand has to be clean, so to say. It doesn't have to, it shouldn't have an inhibitory function or any degradation of target. It should be effectively scalpel-like selective to a specific degradation tag. It's also beneficial if there is a negative control available, because that would then allow to effectively control for any other interactions you may have with a small molecule inside the cells. And then we would ideally like to have full on and off control, which can be provided by different means. And we have seen this exciting photo switchable probes that people have been developing. And finally, we would like to use it in cells, and we would like to use it in animals. So the probe or the control ligand has to have excellent properties in vitro and in vivo.
So today we will have the two different seminars by three different speakers, and let me introduce the first seminars entitled "With the Systematic Profiling of Degron Technologies to Enable Tools for Genetic Target Validations." And this work will be presented by Alessandra Ianari, and she is an associate director in the cancer program at the Broad. Alessandra is responsible for leading drug development projects in collaboration with the Center of the Development of Therapeutics, CDOT at the Broad, or through external collaborations. Her work focused also on the generation of new chemical biology tools to validate new targets and to advance small molecule leads. Her current interest is to use the Degron technology for in vitro and in vivo target validations and toxicology studies. Prior to that, Alessandra worked in the Center of Pharmacology group at the CDOT and at the C4 Therapeutics on protein degradation. And our second speaker, Daniel Bondeson, he's a postdoctoral associate in Todd Golub's lab. And Daniel's graduate work in the lab of Craig Cruz at the Yale focused on targeted protein degradations, where he published on the utility of mechanisms of novel degrader molecules. He currently works on understanding and validating novel cancer vulnerabilities using targeted protein degradations and other novel technologies. His particular focus is on phosphate dysregulation in ovarian and uterine cancer as a novel therapeutic modalities. Thank you very much for agreeing to present your exciting work here. Yes, and the floor is yours.
Hello, everyone. Thank you, Mikołaj and Radek, for the very kind introduction, and thank you for inviting us here. It's a real pleasure to have the opportunity to share on this platform some of the work that we've been doing abroad to facilitate the use of Degron technologies. So if you can advance one, then yes, as Radek introduced briefly, Degrons are protein tags that enable small molecule control over fusion protein levels. And over the past few years, a number of different technologies have been reported, and shown here on the right are just a few of them, organized here in alphabetical order. You can see the plant-derived AI dioxin system, the FKBP12-based Shield and D-tag, the IKZF3 mini Degron, Hello tag, and Smash. And for the most part, these technologies function by hijacking the endogenous proteasomal degradation machinery, which allows a temporally controlled and fine-tuning of protein levels, and in that perspective, clearly differentiating from CRISPR and shRNA technologies. So we, and surely many others listening here, we wonder whether we could leverage this system for target validation and toxicity studies. And indeed, following the identification of an optimal Degron tag, any advanced one, yeah, for any given target, one could envision attempting to determine the therapeutic index that follows target inhibition. And this would be particularly powerful if possible at the very early stages of drug discovery, particularly for novel targets that are not well characterized. Theoretically, this is a straightforward process, which involves the parallel generation of a cancer cell line of interest bearing the Degron fusion target that can be used for efficacy studies in xenograft models, and then in parallel, a mouse model where the Degron is knocked in at the endogenous locus of the gene of interest and expressed as a fusion protein across the whole body. This system could be used for toxicity studies. At this point, we could potentially infer the therapeutic index based on the differential between the therapeutic dose and the dose limiting tox. And while theoretically this whole idea sounds great and on paper easy, our experience abroad hasn't been quite like that. Can you? Yeah. So this is just the identification of the optimal Degron tag. If you can advance one, it's a process that is much easier said than done. It is difficult to, at least at Broad for us, we found that it's unclear at the very start of such a project which Degron to choose among the many different technologies, where to clone it, whether, the N and the C terminus of our protein of interest, which promoter to use that will actually reach the exact expression level that is needed to phenocopy the function of the protein of interest. And even if we are successful in kind of identifying a very optimal, efficient Degron tag, at the end, we have found that Degron tag can disrupt protein function. So with those observations in mind, we wondered whether, well, first of all, there are many different technologies, and one question we had in our mind was whether one was consistently more robust than all the others across targets. And along this line, whether we could develop a system to kind of answer this question and or to provide a system to systematically profile all technologies for any given target and expedite the process. If you could advance one. Yes, along this line, I'd like to present here a couple of examples from our own experience. Here, we're trying to generate a Degron fusion E3 ligase. And for this project specifically, we needed to develop a construct that will lead to very rapid and complete degradation of the target. And so as shown here, we started off by generating a Degron tag construct cloned at the C terminus of our protein of interest under the control of PGK promoter, which didn't work. We then tested the IKZF3 mini Degron, which also didn't work, and then switched termini, placing it at the N terminus. This time we observed very nice degradation, but also unfortunately the appearance of a second band which was not well defined. And it was only 8 to 9 months later when we landed on the AID auxin system that we were able to see clear, complete, very nice expression under the CMV promoter and clear degradation at a very early time point. So it took us 9 months from the beginning of end to just be able to generate the reagent to start the experiment that we wanted to start for this specific project. In another example on this next slide, here it's PRMT5. We wanted to generate a Degron PRMT5 fusion protein. And in this case, we were pretty lucky early on to find that the Icaros PRMT5 fusion construct was efficiently degraded by treatment with pomalidomide. And if you advance one slide, it appeared to be functional in that it was able to communoprecipitate with the PRMT5 obligate binding partner WDR77. And if you advance one more slide, it appeared also to be functional in that it was able to rescue PRMT5 loss, here shown here in single cell clone. And for those of you who are not familiar with the pathway, PRMT5 is an essential gene. So we were very excited to see that we were able to generate this single cell clones expressing a degradable PRMT5 allele. However, when we then probed for total symmetrically dimethylated arginines, which is the methyl mark that PRMT5 plays on its own substrates, we realized that this construct was affecting PRMT5 methyltransferase function, and in doing so, it was not a system that we could use, and we were kind of depressed at this point and really not excited about the idea of starting yet again the same process with a different target. And instead, we decided to pause for a second and ask ourselves a question: is there a way we can develop a system to screen across technologies such that in a few weeks we can identify the Degron target that we need to clone it to, in our protein of it to with our protein of interest to do the experiments that we need to do. So here we decided then to generate a panel of constructs. We selected 5 different Degron tag technologies. Shown here in this scheme is the AID system, the DTAG, IKZF3, the halotag, and the SMASH. I'm not going to get into the nitty-gritty details of how each technology work. This is a conversation we can have offline if you want, but suffices to say that they differ in size of the Degron tag the mechanism of action, and in the drug, the small molecule that they use to be regulated. We generated constructs that allow cloning this each one of these technology for any protein of interest at the N or the C terminus under a strong or a weak promoter, and onboarded. So we generated, basically we have 5 Degrons, 2 promoters, 2 terminites, a total of 20 constructs. It's a lot of constructs to clone in-house. So to facilitate our work, we onboarded everything with Twist Biosciences. And so now, we can just, with one click, we can have our ORF of interest cloned across the whole panel. And in a few weeks, get back the reagents that we need. So we were able to share the panel early on with various members of the cancer program community and with broader large. And that, sharing the reagents with the community allowed us to gather pretty quickly a fair amount of data from 15 different targets, which Dan will show you in a minute. And shown here are people that contributed significantly to this work. I'd like to thank in particular Zachary Muhlenberstein. This is a research associate in the Sellers Lab, now moving on to grad school. Extremely talented, and if it wasn't for Zach, a lot of this work would have not been possible, so thank you. And yes, then you can take it.
Thanks, Alessandra. I'm very grateful to be here, so thanks to the organizers for this opportunity to present our results of profiling these different Degron technologies. So to start off with, we obviously wanted to validate that this Degron system was actually working. And so we cloned a reporter protein nanoluciferase into the vector system and stably generated 293s expressing these Degron tag nanoluciferase. And we found robust expression and, as reported, nice degradation upon the addition of the drug. We'll come back to these differences in expression levels, but just to highlight that our weak promoter or a strong promoter gives very different levels of expression, which can be very valuable for functional studies later. So then we wanted to develop a system where we could pretty robustly characterize the degradation efficiency across these different Degron tags for the 15 unique targets that the Degron working group was interested in. We came up with a fairly standardized pipeline that could take us from about 8 to 10 weeks from clicking on Twist to the final Western blot for all of those constructs. On the right, I'm showing an in-cell Western readout that we adopted for generating dose response and time course data, where we're seeing, for example, that the DTAG N construct for nanoluciferase is not being degraded with any dose of drug, whereas the DTAG C construct is really efficiently degraded at 6 hours with any dose. And we always wanted to evaluate that the full-length protein of the desired molecular weight was being expressed, and then obviously degradation, and then later whether or not this Degron tagged protein was actually functional. So here is the summary of all of our results across the panel. Here I'm showing a heat map for all 15 targets that we had cloned in, binned by the class of the protein. So we have cytosolic, nuclear, plasma membrane proteins. We're showing results just for these 8 different constructs, 4 technologies. We're currently excluding the AID data because of some technical glitches that it's not quite ready for prime time. And here I'm showing the expression level for a particular Degron tag shown as the size of each dot, and the color encodes the degradation potency that we see upon addition of the drug. Just one quick comment on expression, that most constructs were very efficiently expressed, but we did find some surprises where particular proteins like this multipass protein really had very little expression for most of its constructs, and it was only a few that really popped out. Halotag N and DTAG N constructs were generally expressed the highest, which could be a bad thing or a good thing depending on the target. And this might be consistent with some molecular chaperone type mechanism for those N-terminal constructs. For degradation potency, we found that there was a wide range as expected. For example, the cytosolic protein 1 was degraded quite efficiently. Pretty much any construct that we threw at it, it was efficiently degraded. In contrast, this multi-pass transmembrane protein number 2 really only had a few tags, this D-tag N, halotag N, and the SMASH N constructs that were efficiently degraded. And this could be indicative of additional machinery or additional steps or processes that are at play for multi-pass transmembrane protein. Nevertheless, by testing multiple strategies in parallel, every single target that we tested had at least 2 tags that achieved greater than 75% degradation. So how did the different technologies stack up against each other? Here I'm comparing the degradation potency on the y-axis, binned by each degradation technology, where each dot is an individual target. And what we generally see is that there was no technology that really consistently outperformed all others. However, most of the technologies seem to work pretty robustly, at least at the 75% degradation benchmark. You can see that most of these technologies achieved that kind of benchmark pretty consistently. However, when you bump it up to the more important kind of on-off switch, we saw that there was definitely a drop-off when looking at this cutoff. In kind of summary, we found that if you were to only choose one Degron tag, say your lab mate tells you that the Aiolos N construct is just the best thing ever, just choosing only one Degron tag is unlikely to be the most potently degraded for any given target. And also having multiple Degron tags moving forward towards functional studies can be really, really valuable. So we could also evaluate kinetics. As was introduced, the ability to rapidly deplete your protein of interest is really valuable. And so looking at the kinetics of degradation after drug addition across all of our targets, we find that many of these technologies work as reported. This was not a surprise that we could see rapid degradation within 6 hours, and in some cases where we evaluated it, almost complete degradation at 45 minutes. But there were still some surprises where, again, these tricky transmembrane proteins, this is a transmembrane protein here, which was very slowly degraded, and you had to look 72 hours or longer in order to actually observe full degradation. And outside of transmembrane proteins, there were some surprise proteins where this light purple line is really rapidly degraded with other technologies. However, with just the D-tag construct, for example, it was a little bit slower of degradation. So another example of where having multiple tags can be really valuable. I'll make one note on the kinetics of Smash, which I'm sure you can notice. This technology is quite a bit slower, and that's expected given the unique mechanism of how Smash works. The Smash tag is a self-cleaving Degron, and so under normal conditions, you have essentially your wild-type protein with only a small peptide from that cleavage event. This can be an extremely valuable tool for further functional studies. However, addition of the drug blocks that self-cleavage event and requires the relatively slow turnover of that wild-type protein. So that's why it has a little bit slower kinetics. So we think with this system, we developed a way to really rapidly identify just an allele of your protein that could be degraded. But really the important question of functionality has to come next. So although it kind of takes 8 to 10 weeks to really assess degradation, functionality experiments are going to be incredibly critical for whatever protein is being studied. And these are often very, very target-specific assays that need to be developed. So I'll just show 2 examples about those kind of target-specific functionality experiments. This first example is an undisclosed protein where we have a cellular assay for the protein activity. You can see that we can detect that activity in parental cells and then stably knock out the protein of interest and see a nice decrease in that activity, which can then be rescued by re-expression of the wild-type protein of interest. So this afforded a nice system where we could then test the re-expression of the different Degron tag constructs and evaluate whether they're functional or not, and also evaluate the effect of drug. And what we're really surprised to see is that all of these Degron tagged constructs were actually hyperfunctional. Not only did they rescue to the level of wild type, in some cases we were seeing something like 400% the activity of our parental cells. Nevertheless, when you add the degrader drug, we could see a decrease in that activity. Interestingly, it was only the most highly expressed construct that was also the most potently degraded back down to the level of knockout. And so now it's a target specific question again of which one of these technologies is going to be the best case scenario for you. Are you more interested in having kind of the near wild type levels that might not be most potently suppressed all the way down to the knockout level? Or are you really interested in achieving that knockout level of degradation and you're willing to tolerate that high level of expression? I think moving forward, we're still evaluating which of these situations is the right one because it's a fairly novel target where the biology isn't fully parsed out. As a more second concrete example, we at the Broad have a strong interest in MCL1, as I'm sure many of you do. And to just broadly oversimplify the biology that we're looking at, BCL2 family members are anti-apoptotic mediators of cell survival. And we were interested in developing Degron tags for MCL1, and we thought we could leverage a system where the FDA-approved drug navitoclax would inhibit BCL-XL, BCL2, and BCL-W, leaving the cells in this apoptotic primed state, where now if we further inhibited MCL1, this would lead to rapid apoptosis and cell death. And so we asked whether we could knock out MCL1, stably re-express the Degron tagged MCL1, and in the context of navitoclax treatment, the cells would be able to survive if the Degron tagged was if the Degron tagged MCL1 was functional, and then then further degradation of that Degron tagged MCL1 would lead to cell death. So here I'm showing just a confirmation that we were able to knock out endogenous MCL1 and re-express or overexpress, I should say, either PGK or SFFB strong promoter constructs for MCL1. So these were the tags that worked the most potently for degradation, and these are expressed in the A375 melanoma cell line. So I'll take you through the results for our DTAG constructs first. So what we're looking at is a cell growth curve, cell growth on the y-axis over the 48-hour experiment after exposure to the drugs. You can see that our DMSO treated cells are growing just fine, but when we treat with navitoclax, there's a decrease in viability, which is indicating that the Degron tagged version isn't fully functional. It isn't fully protecting the cells from navitoclax induced cell death. Nevertheless, treatment with DTAG V1 seems to almost fully suppress the cell growth, indicative that there is still a window where this DTAG construct is active. So we can crank up the promoter strength a little bit by switching to our SFFB construct, and we see full rescue comparing DMSO and navitoclax. So the Degron tagged MCL1, when expressed at a high level, is protecting cell health. However, treatment with the DTAG V1 compound doesn't fully decrease cell viability. And this is consistent with what we see by the Western blot, where our PGK construct, the weaker expressed that doesn't rescue, only has a pretty slight expression of the V5 tagged MCL1 construct, even though it's completely degraded and leads to a full cell kill. In contrast, the more strongly expressed DTAG-C construct is too highly expressed and can't be most potently degraded, and still leads to some outgrowth of cells. Nevertheless, there's clearly a path forward. We can try different promoters. You can try, in all of these kind of experiments, you'll need to generate clones, and you can pick clones that might have the ideal expression level of your construct. Nevertheless, we had additional Degron tags that we could test in this context. And so here I'm showing the results from our internal Smash tag constructs. For the PGK construct, the more weakly expressed, it's the same kind of story as the DTAG, where weak expression of the Smash MCL1 protein didn't rescue cells and didn't protect cells from navitoclax-induced cell death. However, the more strongly expressed protein really gives the ideal situation where the protein at baseline, or MCL1 at baseline, is protecting the cells from navitoclax-induced cell death. And then we can, in this case, we actually pre-treated the cells with Asunabrevir to get rid of the Degron tagged MCL1, and we see a nice inhibition of cell growth. And I'll just note that this is likely outgrowth of the endogenous MCL1 that doesn't have the Degron tag. So just to kind of summarize our thinking of what makes a good Degron tag, there are a few failure modes that will happen when employing a Degron strategy. If you imagine you have a particular activity that you're hoping a Degron will allow you to toggle between a wild type and knockout, we've seen that certain targets are too highly expressed, such that addition of the drug, even if there is some meaningful level of degradation, it doesn't bring you back down to the level of knockout. So this could be that your construct is just not efficiently degraded and you need to move it around to achieve better degradation. Or it could be that your baseline expression is just too high. A second situation is where you have insufficient function with your Degron tag. Perhaps the tag is interfering with activity, or perhaps your just baseline expression is too low. So it doesn't matter what happens after you add the drug if you're at baseline, you're not actually rescuing the wild type activity. Nevertheless, we feel that testing multiple strategies in parallel is a good way to get to this ideal situation 3, where your Degron tagged protein is fully rescuing the wild type activity, in addition of the drug can allow you to toggle between the wild type activity and a knockout activity. So we think this is a really nice toolkit, and we're excited to share it with the community. So if you can scan the QR code, or I think we'll post a link in the chat to just sign up for updates as we are hoping to onboard gateway compatible plasmids on Agene fairly soon, as well as future publications from this work, as currently this is all unpublished. So we're excited about expanding the toolkit and incorporating additional technologies. Obviously, in vivo applications is going to be incredibly important, whether different tags and the different compounds available for those tags have either durable responses, tissue-specific degradation. These are kind of things that I know that the PROTAC field is thinking a lot about. And in contrast to something like avoiding platelet-induced toxicity with tissue-specific degradation, we're actually interested in addressing these early target toxicity questions. So can we achieve degradation in every single tissue? And ideally have a system where we can have a small molecule that rapidly turns off and then turns on the protein of interest when you withdraw the drug. And this is all leading to this idea of developing knock-in mice, where you can evaluate every tissue upon depletion of your protein of interest. We're also interested in new drugs for new compounds for inducing degradation, so that we can start modulating PK and PD parameters of these drugs to answer some of these important target validation questions. So if anybody has suggestions for that, we'd be certainly welcome to that. So I'd just like to stop there and say thank you to everyone in the Degron working group. Everyone has been incredibly excited about this technology and open to following the standardized protocols as well as sharing their data. So we're incredibly grateful for that. And as Alessandra mentioned, Zach has really led a lot of evaluating a lot of these targets, and Sydney and Tom have also are also RAs that have done incredible work in developing these tags. I'd also like to thank Twist Bioscience for all of the help in synthesizing and evaluating these constructs. So I think with that, I'm going to stop and we can take a few questions before Behnam's talk.
Great. Thank you very much for this extremely beautiful summary of those different tag technologies. It's truly the broad type of approach to the problem. Let's test all of them if possible. So majority of the questions we'll take at the end, but maybe one emerging question is like, how much does the size of the tag really affect of the efficiency of the drug-induced protein degradations? And do you think, taking into account those 5 tags that you checked, how does really compare into efficiency of degradation of the targets?
I think in terms of efficiency of degradation, I don't think we've seen general trends. But for those important functionality questions, certainly our intuition would be that a smaller tag is going to be better. Most people will incorporate epitope tags into their proteins without much of a problem. So I think it might be a little bit more target specific, and it's hard to draw kind of broad generalizations about the size. I don't know if Alessandra, you have anything else to add there.
Yeah, no, I completely second Dan's opinion. It's hard to draw a conclusion.
Okay, perfect. So I think we can move on to our next speaker. Thank you so much, Alessandra and Daniel, and feel free to answer some of the questions in the chat. I think they will continuously appear as we give a talk. It's my great pleasure to introduce our next speaker, who will be talking about protein control using the DTAC platform. And that is Dr. Behnam Nabet, who is now an incoming assistant professor in the Human Biology Division at the Fred Hutchinson Cancer Research Center. And he will open his lab in September 2021. So look out for that. And I'm sure Behnam will talk a little bit about it. He's currently a Catherine Logar Peanut Fellow in the laboratory of Dr. Nathaniel Gray at the Dana-Farber Cancer Institute, and he received his PhD in cancer biology from Northwestern University and a BA in biology from University of Pennsylvania. And in his postdoctoral research, Behnam developed a generalizable technology platform known as the DTAC system to rapidly degrade any target protein. And the DTAC system pairs potent small molecule degraders and versatile tagging strategies to achieve selective degradation of divergent proteins. And the technology facilitates biological explanation and drug target validation in cells and also in animal models, which I guess, Behnam, you will be mentioning. And Dr. Nabet is currently deploying this DTAC platform to investigate pancreatic cancer dependencies. And he has been sharing this technology broadly through various platforms such as AdGene. And Behnam has been recognized with several scientific honors, including the American Cancer Society Postdoctoral Fellowship and Claudia Adam Barr Program for Innovative Cancer Research. So Behnam, without further ado, please take it away.
Great. Well, thank you so much, Radek and Mikołaj, for the kind introduction and the invitation to be able to participate in this really exciting workshop. It's really a pleasure to be joining you all virtually. It's quite exciting as I move on to start my own lab at Fred Hutch in a couple months and really cap off my time here at Dana-Farber, participating in such an amazing series. So I really appreciate it, and thank you again for the opportunity. So I'm really excited to be joining you today to share with you a bit more detail on our development and thoughts around the DTAC system specifically, to give you a little bit more insights into some of the technical considerations that we had in the development of the platform, how we're continuing to leverage it, and some of the key controls and strategies that we've been using along the way. So hopefully this will be a nice complement to the really excellent presentation from Alessandra and Daniel that you just heard. So what I wanted to just frame a little bit is how the DTAC system actually works. And so in terms of the different Degron strategies that that were mentioned, they operate a little bit differently. And so in our case, what we're quite interested here are the bifunctional degraders, or the PROTAC molecules. And we've been, of course, quite captivated and really excited about the potential of targeted protein degradation to really have rapid chemical control over protein levels through co-opting different E3 ubiquitin ligases. And I think we're quite excited about these approaches because they give you both immediate and also, as I'll show, reversible control over protein levels, and they enable you to target the entire protein. In addition, a lot of these strategies are incredibly selective. They give you different pharmacology and responses compared to small molecule inhibitors, which are really fascinating to tease out. In addition, I think it provides an opportunity to really start to think about perhaps overcoming mechanisms of resistance with some other targeted agents, and also new opportunities to tackle really challenging drug targets, which is really the focus of the work that we've been doing here. And so just as a quick reminder, building off of presentations before from Craig Cruz and my mentor, Jay Bradner, what we are interested in are these bifunctional molecules that can engage a target protein of interest and then recruit an E3 ligase such as cereblon, leading to the ubiquitination and degradation of that target protein of interest. And one of the things that I wanted to point out, despite these really exciting advantages that I just mentioned, is that many PROTAC or degrader-based campaigns always begin, or most commonly begin, with an available targeting ligand. So there is something, in fact, that can bind the target protein of interest, allowing one to then create the bifunctional molecule to then see if one can trigger the degradation. However, the majority of the proteome currently remains unliganded. And this was one of the considerations that we had early on as we thought about a lot of these advantages that have been mentioned in creating new Degron strategies and being able to control proteins in other ways. We thought that perhaps we could kind of combine a lot of the chemical excitement around the field with some genetics as well. And so in terms of the rationale around the DTAC system, what we've been aiming to do is really to try to develop a versatile platform that we can continue to build upon that will enable us to really rapidly and reversibly degrade any target protein. And as was mentioned, we were quite interested in developing a strategy that would allow us to study essential proteins, as I'll show some examples for, that would allow us to go after and study in more depth some really challenging drug targets, and would also allow us to start to understand and build medicinal chemistry campaigns designed around trying to actually degrade or inhibit a particular target or protein of interest. In addition, we wanted a platform that would allow us to exploit fast biological responses and would be key for target discovery and target validation approaches. And so in the DTAC system, just a little bit more in depth on how it works, it relies on this mutant version of FKBP12 as the tag. And then what we spent a lot of time developing were these DTAG molecules that can bind this tag, this mutant FKBP12, and then recruit an E3 ubiquitin ligase such as cereblon. This then leads to the ubiquitination and degradation of the fusion chimera. And so what I wanted to do today in the time that I have is try to give two snapshots. One, some of the applications and advances and thoughts that we had around developing the DTAC system. And then in the spirit of the workshop, kind of having a backdrop of some of the technical advances and thoughts that we had and various different approaches that we've used for developing some of the different model systems across various different contexts. And so I kind of want to give a flavor of both the utility, but also a little bit more on some of the technical details that I'm happy to also expand upon in more depth. And so first, of course, I just wanted to give a flavor for how the system works and how we started to assess whether we could achieve really potent, selective, and durable target-specific degradation. So as I mentioned, the first component of the DTAC system, of course, is to use genetic strategies to express your tagged fusion. Similar to the prior presentation, what we initially set out to do was develop strategies that would allow us to N or C terminally tag our target protein of interest with this mutant FKBP12 tag. And also we commonly include other epitope tags such as tandem HA tags that facilitate in the monitoring of the expression or degradation of the target of interest, especially can be quite useful in proteomic studies that we commonly do, but also when antibodies are limiting for your target of interest. The second key component is the DTAG molecule. And so here we were really excited about using FKBP12 because it's been one of the favorite tools of chemical biologists over many years. Back in 1998, Ariad Pharmaceuticals, in a study led by Tim Claxon, published a bump hole strategy around FKBP12. And so what they published was that there's this mutant version of FKBP12, and they designed synthetic ligands that selectively bound this mutant version of FKBP12 and not the wild type version. And so this was an interesting chemical starting point for us because, in fact, we thought that this would be a great way for us to get selectivity over any off targets that we might be concerned about, which include the endogenous FKBP12. And so here, what we set out to do is then design DTAG molecules that would enable us to bind this mutant version of FKBP12, and then on the other end, recruit the E3 ubiquitin ligase. And so now we have a series of compounds where we've been able to achieve this, some of which were mentioned in the prior presentation. And so here, the first one that we heavily invested in was development of cereblon recruiting DTAG molecules. And so here, one of our lead compounds was what we call DTAG 13. And in the process of the development of these compounds led by my colleagues Dennis and Shiva, who were formerly in Jay's lab, what we identified was actually a more selective version of this AP1867 that we could use. And then here, in terms of recruitment of cereblon, we take advantage, of course, of thalidomide as the E3 binder. And so in this case, DTAG 13 was really our lead compound that came out of our initial description. Since then, I've been interested in expanding the toolbox, overcoming some of the limitations that we were faced with, and also comparing and contrasting recruitment of different E3 ligases in different contexts. And so we focused heavily on developing a really highly selective compound that can recruit VHL as the E3. And so this was also recently published. And so just what I wanted to point out, which in my talk hopefully will also come through in some of the experiments, but I won't be able to show everything that we've done. Some of the key concerns and thoughts that we had around developing our system and where we saw limitations in the field when we started this effort was that we really wanted to develop a highly selective platform. And you'll see some of our proteomic profiling studies that we've done and transcriptional profiling demonstrating that we really have no off-target activity with these compounds when you use them in cells. In addition, we wanted to make sure we were operating through the expected mechanism of action when we're recruiting a particular E3 ligase or co-opting the proteasome machinery. In addition, we were really focused on developing a versatile strategy that would be extensible across multiple different targets, across multiple different cell types of interest that we were really interested in actually studying proteins in their more native context. And finally, we really were interested, as I'll show, in making sure that we had a strategy that had the potential of having applications in mouse models. And I'll show some of our data in that regard as well. So initially, what I'll focus on is some of our lentiviral directed approaches where, as I mentioned, we've developed plasmid-based systems that are available on Adgene that allow one to N or C-terminally express your target of interest. What we first actually started with, outside of a number of different controls that I'll skip over in terms of some of our biochemical and cell screening assays that we developed, the first target that we went after, of course, was BRD4, building off of the work that Jay's lab had been doing for many years to target the bet-bromodomain family. And so we were interested in seeing if we could actually selectively deplete BRD4 alone. And so here we did the simple experiment at the time where we cloned in BRD4 with our FKBP tag, expressed it in these MV411 cells, and then used the DTAG molecule to trigger degradation. And we were really excited to see that we could achieve about 95% degradation very rapidly in the cell line of interest. From here, we just started tagging all kinds of different proteins that we were interested in studying in more depth. These include, of course, MYC, EZH2, HDAC1, PLK1, and mutant KRAS. And in each case, we were able to nicely degrade each target protein of interest, confirming some of our earlier observations. But also, we were just really excited because now we were able to continue to build upon this approach. We saw interesting differences in kinetics of degradation, perhaps with mutant KRAS being amongst the slower to degrade, which we routinely see across cell lines. But in general, we were quite excited to see that we could achieve this nice degradation across multiple different targets. So what I wanted to do now with these examples in hand, really hone in on mutant KRAS as an example that I've invested in heavily over the past several years, developing tools that allow us to study rapid biology as a consequence of degrading KRAS, being a currently undrugged target in terms of the mutations that I've been looking at in pancreatic cancer models. In addition, how we've been leveraging the strategy to discover and identify and validate different drug targets, again, with the backdrop of some of our lentiviral directed approaches. And so, of course, mutant KRAS doesn't need much of an introduction, but it is the most commonly mutated oncogene in cancer. It is a driver of pancreatic cancer, which has been a lot of my more recent interest. About 90% of patients present with a most commonly a G12D or V mutation. KRAS, of course, regulates a number of crucial signaling cascades, the RAL-GDS, MAP kinase signaling, or AKT signaling pathways, and has been a sought-after drug target for many decades. And it's been quite a major challenge in terms of developing strategies to directly target mutant KRAS. There's been a lot of excitement and really amazing progress in the covalent targeting of KRAS G12C, but currently some of the other KRAS mutations are unfortunately still undrugged. So what I was interested in doing early on was leveraging the biology that we knew about KRAS as a means to vet the DTAG system and also start to use it to understand and explore the potential of targeted degradation strategies around oncogenic KRAS. And so here early on, starting with a model system where we could take mouse fibroblast cells and then transform them with an oncogenic KRAS. And so this was one of the first experiments that I had done just to confirm the functionality of our oncogenic KRAS, doing comparisons to untagged KRAS versus tagged KRAS, and really seeing if we could elicit the expected effects. And here, gratifyingly, what we saw was increased MAP kinase signaling as shown here, increased AKT signaling as you would expect. And then importantly, upon treatment with our DTAG molecule, we could trigger nice degradation, about 50% within 1 hour. And this was sufficient to actually lead to a collapse in phosphorylation of downstream signaling within 1 hour, which was quite striking to us. In addition, we could do a lot of biological characterization, one example of which I'm showing here, where we saw an increased proliferation in response to introduction of mutant KRAS. This was observed with an increased number of cells in S phase, for example. Upon treatment with the DTAG molecule, within 24 hours, we could completely revert the cells back to cycling like the control cell line. And in this case, also, just as a comparison, we're looking at a control parallel cell line, making sure that we're not altering signaling or any of the biological effects with our DTAG molecule. And so from here, with these systems in hand, then we could really do a lot of rigorous characterization. So what I wanted to show was some snapshots of some of the temporal profiling that we did that I thought was actually quite illuminating and gratifying. The first, of course, was a proteomics experiment. So we wanted to not only look at the selectivity of our system, but also see how the proteome was changing over time as you degraded KRAS. And I'm just showing a snapshot here. There's more detail in our publication, in part just to emphasize how selective our system is, where the tagged KRAS was the only significantly depleted protein in the proteome. We didn't degrade the wild type endogenous KRAS or wild type FKBP12, for example, as shown here. In addition, this then allowed me to couple this with a phosphoproteomics experiment because we had such rapid degradation that we were able to see, as well as potentially, as far as the Western blotting, changes in downstream signaling. So here we saw diminished levels of ERK1 and ERK2 phosphosites. And in addition, we saw a number of other chromatin and transcriptional regulators that were rapidly altered, at least at their at different phosphosites upon degradation of KRAS. And so this was quite interesting to us. We did a little bit more digging and coupled this then with an RNA sequencing experiment. First, to confirm that our DTAG molecule did not alter any transcripts in the control cell line. And then second, upon triggering KRAS degradation, what we were able to do was flip the aberrant transcriptional program that was elicited in response to oncogenic KRAS. The other key experiment, and part of this that I was excited about, was then I could actually combine this and evaluate how does KRAS degradation, for example, compare to targeting MAP kinase signaling? So here using the clinical candidate trametinib, which is largely ineffective in pancreatic cancer models, comparing responses to KRAS degradation. And we saw high concordance, but also quite some striking differences that we're continuing to follow up on. So from here, that was all done in a mouse fibroblast cell line, confirming a lot of the expected biology and really giving us a grasp of how the system was working. What I wanted to do next was then turn to pancreatic cell line systems that we were really interested in further evaluating the consequences of KRAS degradation. And so here, as one example, leveraging our lentiviral system, what I wanted to show were effective means where we were able to express our tagged KRAS, and then at the same time, through using CRISPR-Cas9 gene editing, knock out the endogenous locus while sparing, of course, the tagged KRAS. And so here we have largely consistent levels to our endogenous KRAS as shown here. In addition, we also built some additional controls. So what I was interested in doing was expanding our repertoire of constructs that we could use here, for example, developing a LAC-Z fusion that allows us in parallel to be degrading a target of interest and ensuring that our system is operating as selectively and specifically as possible. Just briefly on the LAC-Z side of things, what I wanted to highlight was that we've been heavily invested in just trying to expand the DTAG platform. So as Radek mentioned, we think it's highly important to have control compounds that are leveraged at the same time. So here for our lead VHL recruiting compound, as well as our cereblon recruiting systems, we've developed these bifunctional control compounds that can bind the tag but not degrade the target. And so that is one example of how we've been leveraging the system. In addition, systems like the LAC-Z construct allows us to, again, do proteome-wide assessments to confirm selectivity in the context that we're evaluating here in a pancreatic cancer setting. And so here's an example showing of again, this time with our VHL recruiting compound, that the only significantly depleted protein of interest is our LAC-Z fusion. So just a little bit more on some of these KRAS systems. So of course, then after engineering the systems, as I mentioned, we could achieve really nice degradation of mutant KRAS. And also, quite strikingly, we could really rapidly depress downstream MAP kinase signaling, as shown here, with reduced levels of phospho-ERK and delayed effects that we could then see on phosphorylation of AKT. And so these types of temporal evaluations are ones that would not be possible really with a genetic approach, especially in a context where genetically we know the cells will die if you knock out KRAS. And here we can confirm this in a viability experiment just 3 or 5 days after treatment with the DTAG molecule, where we see nice loss of viability over time and can do a lot of different control experiments as well, showing that the DTAG alone has no effects on that particular biologic output. And so this has been really quite powerful for us for understanding and looking at the consequences of KRAS degradation in this context, and also looking more in terms of trying to identify targets that are collaborating or cooperating with mutant KRAS, and also potential mechanisms of resistance that might emerge to directly perturbing KRAS itself. And so just to give the reference and a highlight and a shout out, one of the observations that I had was this understudied kinase known as DCLK1 seemed to be operating downstream and collaborating with mutant KRAS to potentiate pancreatic cancer progression, teaming up with Fleur Ferguson, a former colleague in the Gray Lab, who's now moved on to UCSD to start her own group. What Fleur was able to do was develop a really highly selective DCLK1 kinase inhibitor that we could then evaluate in such systems and really start to investigate the roles of DCLK1 in KRAS-driven malignancies. And so that was quite exciting and a really great observation that we initially had using some of our DTAG systems. In addition, in case audience members want to just take a screenshot of some of the ways that we've leveraged our lentiviral approaches, I just sort of wanted to annotate how we've tackled a number of different targets of interest in diverse contexts. These include transcription factors, kinases, scaffolding proteins, isomerases, fusion oncoproteins, and so on. And so in each case, we've been leveraging our lentiviral systems, applying them across different contexts with different molecules to evaluate the consequences of degradation and really study fast biology. So next, I wanted to switch gears and briefly talk about some of the knock-in approaches that we've been using over time and how this has been quite powerful for us in terms of discovering and validating different targets of interest. And so here, what we, of course, again, started with going back to BRD4, we thought it would be a great target to see if we could actually achieve knock-in, but also start to address some questions that we had. And so as Jay had previously shared in his seminar, the DBET molecules, for example, target BRD2, 3, and 4. And so what we were interested in doing was seeing if we could leverage the DTAC system to actually selectively deplete just one BET bromodomain protein and then compare that to the consequences of hitting the entire family. And so what we decided to do was take advantage of the pitch approach, which had been published several years ago, and see if we could start to apply that towards tagging the endogenous locus and tagging BRD4 at its end terminus. And so the schema of what we achieved is shown here, where this strategy takes advantage of microhomology mediated end joining, relying really on small microhomology mediated domains to be able to achieve knock-in at the BRD4 locus. And again, plasmids and protocols that we've shared are available on AdGene for this approach. So here in 2932 cells, we were able to achieve this and develop a homozygous knock-in clone where we could nicely tag BRD4, trigger its degradation with the DDAG molecule, and sparing BRD2 and BRD3. Here then we could do the comparison where we evaluate the pan-BET bromodomain degrader, DBET6, which hits BRD2, 3, and 4 quite selectively and potently, and compare that to the consequences of BRD4 loss. And so this was a way that we can start to then mechanistically dissect and deconvolute perhaps what contributions BRD4 might be having compared to pan-BET bromodomain degradation or inhibition. And so one snapshot, just to give a highlight of what we were able to observe in this context, was that really there was quite modest and surprising to us effects upon degradation of BRD4 alone when we developed the homozygous knock-in clone. Here, in comparison to pan-BET degradation, as shown here in black, or JQ1, June's BET bromodomain inhibitor that hits BRD2, 3, and 4. Again, we saw quite interesting differences in the pharmacology of these different agents. And so this is something we've been continuing to evaluate and looking at the different contributions of the BET proteins over time. But I think a nice way that one can really leverage systems like the DTAG system to really deconvolute and start to evaluate agents that might have activities on multiple different proteins and see what the contributions of each factor might be. Now, in terms of knock-in approaches, in that case, we used the pitch approach, and I just wanted to highlight again, in case audience members were interested in some of the other strategies and contexts that we've applied this to ourselves. So in collaboration with Rick Young, we tagged YY1 using HDR-directed methods with Peggy Goodell's lab, tagging NPM1 with Georg Winter, mediator subunits, and then with Jeannie Lee's lab, WAPL and RAD21 as targets that we were able to achieve nice knock-in using various different approaches. And I'm really excited to see also the community harnessing this approach and using it in other ways. So there's many different ways that one can achieve expression or knock-in. And so these are just some of the strategies that we've used, but it's great to see some of the emerging technologies that others have as well. And so what I wanted to end on finally were some of our really investment from the beginning in trying to develop a strategy that would have in vivo potential, specifically in mouse models, but we've also been doing work in other model organisms as well. And just giving a more recent snapshot of how we've been leveraging this for evaluation of drug synergy and synthetic lethalities. And so here early on, what I wanted to do was see if we could develop a system that would allow us to do non-invasive bioluminescent measurements in a mouse. And so here what we did was tag luciferase, express it in MV411 cells of leukemia cell line. And here's just the in vitro confirmation that we're able to depress luminescent signal in these cells. From here, we set up the following scheme where we could tail vein inject these cells into mice. After 2 weeks, when the mice have established leukemic burden, they then glow. And then sequentially, we could do treatments every morning and then do a luciferase measurement 4 hours after that treatment. We could do this every day. And on the final day, actually, we didn't do a treatment. We wanted to look at reversibility and kinetics of recovery. And so this is effectively a 28-hour time point where we could then do another measurement. And so I had initially done this in our 2018 paper and then went back as we were screening more molecules and comparing cereblon versus VHL mediated degradation and repeated the experiment. And so here what we were, just to show you what these mice look like, we see that they nicely glow when they've established leukemic burden. So we're not controlling tumor burden in this experiment, just looking at luminescence, loss of luminescence as an output for effective degradation. And so here, hopefully what you can appreciate is that we can significantly depress luminescence signal upon treatment with our VHL recruiting DTAG molecule. In addition here, then you can quantify the whole cohort of mice. Here, the comparison was between DTAG13 and DTAG V1. In each case, we do see really robust degradation. Again, this is 4 hours after an administration that we're doing the measurement. On the final day, what we had observed initially was that there was recovery with the DTAG13 ligand, and what we were hoping to improve on, which we did with our DTAG V1 molecule, which has improved PK properties, we do see improved diminishment over time, suggesting that perhaps different dosing mechanisms can be used when employing these molecules, but it needs to be worked out in further detail. And the last bit of data that I wanted to show was a paper that we had that just came out with the Arribas lab this past month, where they had identified that SLUG was a key transcription factor that coordinated resistance in pancreatic cancer to MAP kinase inhibitors. And so here, this is shown in an in vitro experiment where you express tag SLUG, for example, and you have a decreased responsiveness to the MEK inhibitor. This can then be alleviated when you combine this with SLUG degradation, as shown here. What we then did was a short-term xenograft experiment where we implanted these MIAPACA2 cells into mice. And what you can hopefully appreciate is that the single agents alone, SLUG degradation or the MEK inhibitor, really have little effect on tumor burden. And here the combination synergizes quite nicely in this really acute experiment. And so this was one of our first examples of achieving nice degradation and looking at a synergistic interaction in a xenograft mouse model. And so this is just a bit of data that I wanted to highlight. And so hopefully today I was able to share with you a little bit on our development and thought process and controls around development of the DTAG system, some of the ways that we've been leveraging it for fast biology, discovery and validation of targets, looking at drug synergies. In addition, some of the technical approaches that we've been, and milestones that we've reached and things that we're continuing to evaluate and build on, looking at lentiviral, knock-in, and in vivo applications. And so as Radek mentioned, I've been really interested in trying to make this as open and as accessible as possible for the community. As I mentioned, plasmids are an AdGene, molecules are commercially available and also can be provided by us. I've written several protocols, guidelines, and book chapters that hopefully can be helpful as well. And this has just been a great way to also start to collaborate and has led to really a number of different investigations that I've been really excited about, which has been quite amazing. And so with that, I need to thank many people, of course, Nathaniel and Jay for really being amazing mentors over the past several years when I've been at Dana-Farber. Their support has just meant so much to me, especially as I've been transitioning in the past year. Members of the Gray and Bradner Laboratory that were just instrumental in the development of the system and just great colleagues to work with, as well as numerous other collaborators on various different projects, and funding that has supported my time here at Dana-Farber. And so as Radek mentioned, I'm really excited that I'll be opening my lab at Fred Hutch in September. I'm actively recruiting, so if you're interested in targeted protein degradation, oncogenic signaling, transcriptional signaling, developing new degradation-based approaches, reach out to me on Twitter. My new email is listed here, so please send me a note. I would love to recruit and also collaborate. So thanks again for the time and for having me join today. I really appreciate it.
Thank you very much, Behnam, for this wonderful talk, and then a walk through various different developments that you have advanced through the years on the DTAG platform. There are quite a few questions. I think a lot of them have been answered by Alessandra and Daniel, but I think we should come back to quite a few of them. And then there are a few more specifically to your talk. And one that I think is quite interesting is the role of ubiquitination on the tag molecule itself. So the question is basically, do you see the efficiency of ubiquitination on the tag or on the target impacting the speed of degradation, or perhaps it doesn't matter?
Yeah, sure. I can start on that one. I think, yeah, we haven't we've looked at it in a bit of depth, something that we're continuing to work on. I think it is a little bit target and context specific, where we see either tag-based degradation, at least on the FKBP side. There are 8, I think there are 8 different lysines that could be ubiquitinated, or on the target as well. I think early on we thought it was really tag-driven, but in certain contexts where we see different E3 ligases being better suited to degrade that particular target, it seems to be that perhaps there has a lot to do with the target that it's also fused to and the accessibility of those lysines as well. And I think that speaks a lot to your work, Radek, also the structural features that are quite important in mediating a lot of these types of responses. So for us, it's been a mix of both, but we definitely do see the ubiquitination, but it kind of varies where it might be.
Thank you.
Maybe continuing on the license requirement for the tags, how do you think it's like the license on the tags are usually utilized in the linker or in the target of the protein? What do you think about general the requirement of the available license?
Yeah, I think that it's something that we would like to test. So we have started to do it a little bit in terms of developing constructs to mutate different lysines either in the tag or where we might know where it is in the target of interest to start to evaluate that. I think it seems to be a bit context specific, at least in the cases that we're starting to evaluate, in terms of it either being the tag or the target or both. I think that certainly happens as well. So some targets, I think, if we do, and I think it'll be like a comparison that we're thinking about, for example, is one, EWS Fly has been one where we've seen VHL recruitment being as the suitable E3 ligase to be able to degrade it, where we know in those contexts, you can either co-opt cereblon or VHL to degrade other targets that are tagged, for example. And so in that case, we see it's an example where we can start to dissect maybe that case, it's likely the target that is being ubiquitinated because we're using that universal tag that we know can be ubiquitinated normally. But then comparing that to other targets of interest like BRD4, which seems to be degraded in every cell line or any context or any PROTAC that one develops. So looking at more easily versus more challenging targets, I think will be informative in that way.
One aspect that we wanted to add to the panel is generate like a panel V 2.0 where we can also compare the different linkers, because right now the linkers that we are using is a rigid linker that we got from the Eber lab, by the way, and that contains a fair amount of lysines in it. So one of the experiments we're currently running is comparing different linker lengths and rigidity and whether or not they contain lysines. And if we do, we will observe some significant differences, then we'll definitely upgrade the panel to include different options such that you can avoid having a target degraded basally in the absence of the chemical inducer just because it gets ubiquitinated and degraded, or you can promote and boost the degradation for difficult to degrade targets. So, but it's still an open question as far as we know.
Yeah, I definitely agree that it's an open question. I'd mentioned this in the chat, but I'll just say it here too, that in my graduate work with Craig, we were trying to answer some of these questions about lysine preference and lysine selectivity and stuff like that. And we'd found pretty promiscuous ubiquitination across like a halotag GFP substrate. And even if you mutated away some of the most ubiquitinated lysines, it seemed like there were now new lysines that were going to be ubiquitinated. I think it's pretty target and probably E3 specific, but it does seem like some of these E3s are pretty promiscuous once you get a substrate into the ternary complex.
So there's a question in the chat that just appeared, and it's, can your approach degrade both existing and newly synthesized targets? How are proteasomes being protected from being effectively limiting factors in the targeted protein degradation? So I guess in this particular case, I guess if you target with a D tag or with any Degron tag, you effectively degrade proteins, any proteins that are like created, and then they're both newly synthesized or not. But is there a way of learning anything about the resynthesis rates from these kind of degradation experiments? Maybe that would be another question.
Mm-hmm. Yeah, I guess just speaking to that, and I guess Daniel and Alessandra can jump in. I think probably the most distinguishing one would be Smash in that case. That is the one that kind of, in terms of the synthesis with or without the tag as a result of just the mechanism of how that platform works, it's actually quite distinct from all of the other ones. So there is a difference between newly synthesized or resynthesized proteins compared to some of the other approaches. And that's why probably in Daniel's data too, you see that difference, that striking difference in the kinetics of degradation, because you have to wait for the natural turnover of the protein as well.
Yeah, absolutely. We tried to do some experiments to address whether we can gunk up the proteasome or the degradation machinery, where we can transiently transfect different amounts of your protein of interest and then test whether there's a limit to degradation potency. And we do see that. So at massive levels of overexpression, you'll see a complete drop off in degradation potency. Whether that's, what is the limiting factor in those experiments? We haven't really evaluated that, whether it's the drug, whether it's the E3, whether it's the proteasome. But these likely are saturable processes that could get gunked up.
Yeah, we definitely saw that too early on with DTAG, just in like 293 T cells, for example, where you can really do that type of assessment, and it was disheartening at first actually seeing the lack of degradation. That's why we switched, to be honest, to the MV411 cells that I showed that really early experiment where you could tune it a little bit differently and try to match, for example, endogenous levels, and then you see quite distinct effects.
Yeah, and that's also another of the questions that we wanted to answer by comparing across technologies, whether some technologies are easier to saturate than others. Like, we don't know that. We know that the technologies can be saturated at very, very high levels, but we haven't done a systematic assessment for that.
Alessandra and Daniel, you beautifully showed, that really one have to test multiple different Degrons for your target of interest. But would you have any recommendations for choosing which features of the Degrons would you choose for endogenous tagging? And how would you think about this? Because this is even more time consuming. So it's even more difficult to test the panel of the Degrons. So what would be your kind of like general criteria to think about endogenous tagging of the favorite dependency?
I can jump in, and Alessandra, you follow up. I think if you can be guaranteed ahead of time in that you've actually tested it, that your Degron tagged version is functional, then it's probably worth the investment in trying to knock in multiples. If I were forced to choose and you had multiple options, then going with some of the smaller tags might enable a much easier knock-in route where you can use an RNP rather than the CRISPR plasmid approach. But as Behnam nicely showed, the CRISPR approach seems to be really, really efficient, and you can design it well. So I think the smaller tags might be a little bit easier, but that plasmid approach seems to work pretty well.
One other thing I was wondering about, looking at like your talk, Alessandra and Daniel, was the striking difference between N and C terminal tagging in terms of the protein expression. So you could see much higher expression of the protein with N terminal tags, regardless of the promoter. But I think it also was a little bit counterintuitive, because the higher the expression of the protein, I think the better degradation was observed with some of these tags. Is that something that you have seen? Is there an explanation for that? Or like sort of?
We tried to do some correlation plot between efficiency of expression and efficiency of degradation, and there wasn't a clear correlation. Maybe sometimes, yeah, the bubble plot could be visually confusing in that regard. There is no clear correlation that we could detect, and likewise, it wasn't that the N terminus was expressed better than the C terminus. It was really on a target by target basis and on a Degron by Degron basis. Well, mostly on a target by target basis. Certain targets could not be tagged at either one or the other termini, period. Yeah, so if you know that ahead of time about your target, then you can skip half of the one altogether.
How do you see the future directions for the optimization of those methods? Do you think there is a need to have more of those different Degrons, more different drugs, or do you see more that we should optimize those Degrons, that they should be more efficient? The ones and the set that we have already is potentially sufficient.
I would take a stab and say that we might have a good set already, and that just in terms of the raw technologies, it seems like what's out there will be able to achieve degradation for your protein of interest. I think that as Behnam nicely showed, having new compounds is really valuable. So having the ability to tune half-life of your compound for prolonged suppression of your protein, or really rapid and transient on and off, it's kind of dependent on the particular use case. I don't know, what do others think?
Yeah, I agree. More compounds would be great, particularly as the example of the DTAC technology shows. If you Behnam can generate another couple of heterobifunctional molecules that are engaging different E3 ligases, maybe that could also expand the toolkit that we have at our disposal. That could be one idea, but compounds with better PK/PD properties also would be a great addition. And I agree with Dan that, yeah, we have a fair amount of technologies. Actually, we haven't included all the technologies that are available. There are more out there that we're planning to include. So I would, yeah, rather focus on the compounds, and maybe focus on how to generate, if possible, to generate like a systematic approach to perform knock-in of the endogenous locus. That would be greatly beneficial for the community, because everybody is saying that everybody's doing it, but no one talks about how difficult it is to actually get it done from beginning to end.
Yeah, I totally agree on all those points. I think that, yeah, adding to the repertoire, we saw such a huge, it felt kind of minor at the time as we invested a lot on the VHL recruiting compound, but it has made such a difference for us in general, I think, for a lot of the application and some of the target scope that we have accessible in context, that adding, I think it's easier to add a bit more to the technologies as we vet them, if we closely vet them and do the appropriate controls, versus sometimes it's so much work to create a new technology as well. So I'm of course biased, but I think that is, there's so many E3 ligases to co-opt and sort of expand on the platforms and learn just how they behave and differences in vitro. And then as Alessandra mentioned, I think a big barrier for a lot of these technologies to date is in vivo applications, and there's still a lot to be learned, just how well you get degradation in a xenograft model versus a transgenic model, and in what tissues you might get degradation, how that compares to a PROTAC or an inhibitor. Those types of questions are all ones that I hope with our systems at hand are currently at a stage of being accessible to test. And then, yeah, new technology is also working. I think figuring out best ways to combine the existing technologies is something that is kind of exciting too, manipulating multiple proteins at once, and looking for, in my end, synthetic lethalities, drug synergies, and so on. That is an exciting way of just kind of harnessing and having a repertoire of compounds where halotag, as I understand, usually is only right now VHL, and maybe there's an IAP molecule. I'm not sure how well that performs, but VHL driven, combining that with DTAG cereblon driven, having more compounds allows you to do that type of assessment as well for other types of biologic questions. So I think those are areas that could be quite impactful also, as we see for target validation studies towards clinical combinations that we might be pursuing, and ultimately gaining some insights down the line as well that could be useful.
I think Behnam answered my also last questions. Which type of applications but maybe I ask Alessandra and Dani, which type of applications of those Degron tags you feel that are the most exciting for you, and you think in future they will really answer? Where do you really see this technology moving forward? Which, in combination with which other technologies, they will be extremely powerful? I don't know. And Behnam, feel free to add to your answer if you still want.
Who's this question for?
It's for the whole panel. It's yeah. What do you think you?
I think I started my presentation like making a pretty clear statement that I think I mean, at least in the drug discovery field, it would be great if we learned a way to easily transition from in vitro studies to in vivo studies, and really figuring out a systematic way to determine the therapeutic index of any given target, particularly for targets that are novel and not characterized yet, for which we don't have optimized compounds. Yeah, that would be very, very powerful way to de-risk projects very early on. And or to figure out, even if the project has started, even way to figure out which dosing regimen you want to use. Sometimes for certain targets you want to have continuous inhibition. Other times you want to do pulse dosing, and that is something that you can ask with this system, and you can ask early on and guide medicinal chemistry. I think that would be very powerful, but no one has provided proof of concept that you can actually do it. So that's one of the things we were thinking about here at the Broad, the cancer program, to start maybe just generating a proof of concept study and see how to do it.
Yeah, I totally agree. That's, you find a novel target out of cancer genomics or functional genomics or something like that, and you have no idea what the off-target or the on-target toxicities might be, and being able to unlock that would be really valuable. The other nice thing is that you don't have any tools to really study your protein. So having the ability to turn off your protein within an hour, that's amazing compared to CRISPR, where it's 4 days, or SH 2 days with all sorts of off-target effects. Unfortunately, I'm in the situation of studying one of those nasty transmembrane proteins that still takes something like 2 or 3 days to be degraded, but it's slightly better, I suppose.
It's fancier.
Autophagy, I would say.
Yeah, that could be an interesting the LITAC approach. Yeah.
Right.
Thank you very, very much for this great panel, great discussions, and presenting this very exciting technology. To everyone, to the whole community, please let us know probably over Twitter whether you like this type of workshops, whether we should be thinking about different types of topics for future. And thank you very much for answering all the questions and great discussion.
Thanks, everyone.
Great, thanks so much. Yeah, feel free to read their questions that come up later.
Likewise. Thank you. Bye-bye.